Guys. All right, so we have Dr. Jason Spadaro, right? Am I correct?
Yeah,
All right,
yeah.
serves as the manager of Mote Marine, as you guys already know. Restoration research program. His research focuses on landscape scale restoration of the Caribbean coral reef communities. His work is primary focus on in Florida coral reefs. He has a bunch of active projects throughout the Caribbean, including Puerto Rico, Antigua, St. Kitts, Belize, and Mexico.
Oh, thank you.
There you go, sir.
Thank you so much. Thank you. And so just wanted to share with you guys some of the work that we do at Mote here on Florida's coral reef, especially with some of the folks that are helping do that work. So this is Restoring Reefs with Mote Marine Laboratory, Science, Innovation, and Resilience. And so just some context. I think that we're all familiar with coral reefs. Yeah. And coral reefs are one of the most biodiverse and ecologically complex ecosystems on the planet. But interestingly, even though they are one of the seats of biodiversity on the planet, if you aggregated all of the living coral reef space on the planet into one place right now, it would all fit inside of Colorado. So despite housing somewhere between 25 and 33% of all species that occur in our oceans, not the individuals, but the species that occur in our oceans, somewhere between a quarter and a third of them, they only occupy less than 0.1% of the seafloor. Yet still, one of the seats of biodiversity, and because of that, it's all the more alarming that we have, and we should care about the health of these reefs if we enjoy breathing, right? I do. Yeah, everybody does. While coral reefs don't produce a lot of the oxygen that we breathe, our open ocean ecosystems do, and just like the canary in the coal mine, coral reefs are kind of the herald of issues occurring in our open ocean ecosystem. So as we see our coral reef ecosystems start to fall apart and those ecological interactions and complexities break down, we should be paying attention to that, because that's a signal that larger problems are at work behind the scenes. Especially if we don't want clean air to become a rare commodity. When I was a professor, I knew that it was time to stop when no one in the class knew what this was. Does everybody know what spaceball was? Yeah, okay, thank God. Thank you. Coral reefs are also a major economic driver, especially here in the Florida Keys. We're sitting in a very, very good example of why coral reefs are an important economic resource for the Florida Keys. Obviously recreational snorkeling and diving, it's almost lobster mini season. And that's a beautiful example of one of the economics, goods, and services that coral reefs provide, both for tourism and for recreational and commercial fishing. But they also provide us a much more valuable ecological and ecosystem service, and that was highlighted during Hurricane Irma. During Hurricane Irma on 10 September in 2017, just about 8 years ago, we had roughly $58 to $83 billion in economic damages here in the Florida Keys. I don't know how many of us were here during that time. It was like a war zone. I came in right after the storm, and it was awful. And if we had not had that coral reef out there, that damage would have been substantially worse. We probably could have added a zero to the back end of each of those numbers in terms of damage, and it would have been much more catastrophic. And that is because coral reefs actually serve as sort of it's not really a speed bump, really, because they're taking all that kinetic energy coming from offshore during these storm events and redirecting it, redirecting that energy into different directions so it dissipates and does not make it through in the same form as it did before it hit the reef. And we actually have some fantastic, relatively newer data from Eastern Dry Rocks, just off of Key West, where we install, and actually, selfishly, I did not realize this until I looked at it. That's actually me helping Jim Hench from Duke install a bunch of instrumentation off of Eastern Dry Rocks, where we looked at wave pressure coming in from offshore during Hurricane Ian. Happened to be out there just in the middle of this experiment. 97% of the kinetic energy coming in from offshore during Ian was dissipated by the reef crest of a degraded coral reef ecosystem in the Florida Keys. So in other words, only 3% of that energy made it in to impact our shorelines. And I watched that water line come right up to my back door. So it was, we need to definitely pay attention to the structural integrity and the biodiversity of our coral reefs, because they are incredibly directly and indirectly economically valuable to our system. And as they're designed to take on a lot of this kinetic energy and all of these interactions, why then, if they are disturbance-mediated ecosystems, they actually require disturbance to maintain that high diversity condition, just like our longleaf pine systems throughout much of Florida. If you don't have fire in these systems, they change. They are no longer longleaf pine systems. They become something very, very different. So if they're disturbance-mediated ecosystems, and we're definitely seeing disturbance on coral reefs, why then are they in trouble? This is a 10-year challenge. It's a photo of a coral head off of the Great Barrier Reef, and then 10 years later, the same photo. And this was I think it was in Science. Why are we seeing this if they are disturbance-mediated ecosystems and we're seeing disturbance in them? And it's because that natural disturbance regime is exacerbated by a bunch of additional anthropogenic stresses, like historic overfishing, not just overexploitation on a kind of a point source level, but systemic and historic levels of overfishing, acidifying seas, warming sea surface temperatures, as we saw very clearly in 2023 and 2024 right here in Florida, and a couple of other insults that we're throwing at our coral reefs, which lead to things like systemic increasing incidence and magnitude of coral diseases. So this is stony coral tissue loss disease, taken by one of our colleagues right at FWC just a couple of years ago. And you can see that this is a disease lesion. This is stony coral tissue loss disease, the one that's still out here. This is a disease lesion that started in January 5th. And by February 1st, 90% of that colony had died. So this is one of the most aggressive and lethal coral diseases that we've seen. And new versions of these diseases are emerging every year. And they dramatically threaten our biodiversity in the system. And that confluence of stresses, everything from climate change and overfishing and overloving those reefs to disease and nutrient pollution, can take a system, this is Chica rocks, from looking like this to something that is devoid of coral as the dominant taxa on the bottom, and allow for some other critters like sponges, octocoral, and algae that can capitalize on those conditions that coral are really struggling with. So what we call that is a phase shift, where we go from a coral-dominated stable state to something that is dominated by other things. And how our job as restoration practitioners and scientists is to figure out how we take a system that has gone down into the other side of the hill, and how do we push that system back up over those thresholds and into a stable state that looks more ecologically or visually like what was there before restoration. And we do that in a number of different ways. I'm sure that this is a relatively familiar site. Does everybody recognize this? This is among this is Lou Key, and it's our offshore coral nursery. Mote now has 4 of these, one just offshore of Key Largo here. And in it, we focus on branching corals. This is Acroporis cervicornis, or the staghorn coral. Most of that we produce is done in these offshore nurseries. And then most of our massive form, the slower growing, reef building massive form corals, are done on our land-based systems. And we've got 3 of those. One here in Key Largo at Reef House Resort, one at Bud and Mary's Marina, and then the big one that my office is at down on Summerlin Key. And the idea is that we take a small amount of material that we know the genetic identity of, make a lot of very small fragments of that material so that we rapidly produce a large amount of coral to deal with from a relatively small amount of starting parent material. We take those fragments and install them on the reef in monogenetic clusters, and that means that each coral in here is a clone of a single genetic individual. And we do that on purpose with the branching corals and with the massive form corals. Each one of these is a clone of a genetic individual. Because as they grow and they touch, if they're different genetic individuals, they'll actually fight, chemical and physical warfare, and that slows down the growth of either individual. But if they're the same individual, they'll actually grow, touch, and they recognize that they're the same individual, and they'll fuse. So instead of putting out a single fragment and letting that grow very slowly through time, we put out a whole bunch, and that rapid growth during the microfragmentation process is capitalized on, and you get a fused large colony in a fraction of the time that it would take with more traditional kind of fragmentary lifestyles. And the idea is to rapidly increase the cover of coral on the reef, but also much more so, so obviously with the acroporids, you're trying to build a structural element on the reef. So this is a very complex architectural element that is facilitating a number of different species as habitat. With the massive form corals, we are trying to overcome what's called Ali effects. So as you lose corals in our ecosystem, and as we have lost corals on the reef, they can't move, right? So if we go in and we lose 8 out of the 10 moose in a population, they can find each other still, those 2 remaining ones. Corals, if we lose 8 out of the 10 colonies that are able to reproduce, and those 2 that remain are pretty far apart, the probability of them reproducing together drops off dramatically. So the idea is that we want to put in a bunch of other reproductive colonies to link those 2 together and really drive the probability of reproduction back up through the roof. And this is a success story just off of Big Pine Key, off of Cook Island. And these fragments, the fragments that produced this colony were outplanted in 2015. And this photo was taken in 2020. So in 5 years, we went from little microfragments to a reproductive colony in a massive form coral. This has never been demonstrated before. This was the first time, and we're actually just about, if you look at those dates, next week is the 10-year anniversary of these guys getting outplanted. So we're hoping to see them spawn again.
How big would you say that is?
That colony, I was right behind the guy with the light on the other side of it. That colony is about that big.
Okay.
Yeah.
5 years. You said 5 years from these to that.
5 years from 10 or 20 fragments in a thing like this, and it was, Yep, absolutely. So how do we reverse this phase shift? How do we make that happen at a larger scale? So obviously we can do it relatively small scale. You can do that a few places here and there, but the coral reef that we're trying to restore is 360 miles long. How do we make that little success occur over a much larger area on Florida's coral reef? And one of the ways that we do that is that we have multiple different locations throughout the reef. And now obviously Florida's coral reef extends well beyond the dry tortugas out to Pulley Ridge, and then way farther up than Homestead and Florida City. It goes all the way up to Fort Pierce. But to do that, we're just focusing on the Florida Keys National Marine Sanctuary right now, which is about 120 miles from essentially what is Biscayne Bay all the way down to the dry tortugas. You can't do that with one coral nursery, right? So we have, and this is a terrible slide, I apologize. We have a nursery here at Summerlin Key, a nursery here at Sand Key off of Key West, a nursery here at Lou Key, a nursery here at Isla Morada and at Bud and Mary's Marina, nursery here at Key Largo, and a nursery here at Reef House Resort and Marina. And the geographic spread of that production capacity and staff capacity to then take those corals and put them onto the reef gives us the operational environment that extends across the entire coral reef tract in the Florida Keys National Marine Sanctuary. Working on other things going on, and basically that is 3 land-based coral nurseries and 4 offshore nurseries with a combined capacity, if we filled all of them completely up, of more than 2 million coral fragments at a time. We've not ever come close to that because we're constantly putting them out. And we'll show some numbers on that here in a bit. That's not a small job. It takes 26 full-time staff, working every day to service those 7 coral nurseries. And even then, every one of my staff is working very, very hard to do all of those things. We accommodate somewhere between 6 and 24 seasonal internship opportunities every year. We have dozens of dedicated volunteers assisting with that process and numerous partners. The collective goal here is innovating, which is one of our interview questions. Like, what does innovation mean to you? And to me, that means trying to do more with less, or trying to do more with the same, and keep trying to figure out how to make that same dollar, coral, labor hour, so on, piece of material go further and further and further and increase that efficiency of operation, not because we want to bring in more money or anything like that, but because look at the job that we have to do. The need for coral reef restoration, the need for these successes and failures in restoration is growing exponentially. The funding for it is not. So we need to find ways to make every bit of effort that we put into this go further and further and further, and that's our goal. And across that, those 7 nurseries service. This is the 7 1-mile diameter permitted areas that we currently work in. So that is the area that Mote conducts coral reef restoration within the Florida Keys National Marine Sanctuary in. Each one of those red dots is 1 nautical mile in diameter. And over the course of the last, 15 years? Mote has outplanted quite a few, but we came from very humble beginnings. In 2008, we outplanted the first corals onto Florida's coral reef. And it was 112 corals over the course of that year. And then the following 3 years, 2009, 10, and 11, we didn't outplant a single coral. We were still building out a whole bunch of the regulatory framework around how do we get those corals out onto the reef in a way that is responsible, ethical, and appropriate, so that we're not swamping the genetics of the natural populations, we're not introducing disease, we're not doing a whole bunch of other things. And then in 2012, we bumped up. I think we were up to several hundred. 2013, 14, 15, we were increasing dramatically. 2016, we saw a big bump, and then just kind of a slight drop in 2017. I think I already hinted at why. And then 2018 was another drop, and then we jumped again, and then we jumped again in 2022, and we've dropped off a little bit. And then already this year, we're in July now, and we've outplanted roughly 15,000 corals onto Florida's coral reef this year so far. If you look, during this time, despite the fact that stony coral tissue loss disease showed up and ripped through the entire Florida Keys National Marine Sanctuary and the rest of Florida's coral reef on the north end in 2014, from 2014 to about 2019, that disease was running very hot. We still were increasing in our effort. During the latter part of 2017, we had Hurricane Irma come through and do a whole bunch of not just damage to our infrastructure and our offshore coral nurseries, but to the framework of the reef. So Hurricane Irma literally changed the landscape out there. And still, we saw this dramatic increase in effort and success. And then we had one of the most catastrophic, or at least dramatic, marine heat waves in 2023, but it persisted all the way through into October of 2024. And still, we're set. So over the course of that time, we have now currently outplanted more than 258,000 coral fragments onto Florida's coral reef, and we are set to dramatically increase that by the end of this year. This is the projected number of outplants for this year. Through that time, one of the simple innovations that we've made to make that restoration intervention occur at a much larger scale and much more efficient. How do we make every dollar, every piece of coral go further? You don't leave the dock for less than some number of coral. Early on, we were outplanting a few hundred corals. So this is actually 10 years after we started. We're still only averaging about 225 corals per trip of the boat. And I'm sure that everybody's aware that boats are not inexpensive to operate. Staff time is not inexpensive. All of those kinds of things. So the cost per coral was very high. As we move through, that number really starts to dramatically increase And then in 2025, we're up to about 1,100 corals per trip. So all of a sudden, the same material and labor cost is giving us dramatically more corals onto the reef. So it's one way that we can increase how far each of those dollars is going. And still, this transition is very, very difficult. And we've been doing it in a number of ways. It's very similar to trying to restore this. Everybody's seen this before, right? And if we just did nothing to this, something would happen, right? Would it be a forest immediately? Or when you see people go in and restore these areas, are they planting redwoods where they were? No, it's a series of ecological steps. However, in coral reef restoration, we've always gone for, we clear cut an area and let's plant the redwoods right back in before handling anything else. And that's what we've been doing for many, many years. And that is because coral reef restoration takes its roots, literally, from silviculture, from forestry. We even call them coral nurseries, which are really the term that you use for a tree farm. And we outplant those corals and trees in these monogenetic, very ordinal fashion. So you get these, I mean, you can literally look down, and if you drive up 75 through those pine barrens or up 95 on the other side through those areas, if you look at the pine forests, you see these beautiful, exact rows of trees in there. That's not a natural assemblage. Those are literally a farm for wood and pulp and things like that. But we've taken that in coral, and you can see obviously in the nursery, we outplant these things in clusters to get that assemblage, but it was this very uniform distribution of a single species, multiple genetic individuals within those arrays, but this is very not natural, right? However, this was how you got up to this. And because Acropora, the branching staghorn coral, is probably the single most common species used in all coral reef restoration across the globe. Obviously, this species is endemic to the Caribbean and Western Atlantic, but there are analogs to it that are being used in Israel and the Red Sea and the Indian Ocean and the Indo-Pacific, all around the world. Acropora, the branching species of corals that grow fast, they're kind of weedy, and you get this dramatic result in a relatively short period of time. That dominates restoration around the globe. So we've basically treated them as we do trees. And you can see here, this is called a structure from motion, large area imagery file. So this is a whole bunch of still photos that have been stitched together into three-dimensional model and then flattened so that I can use it here on this presentation. And every one of our coral fragments has been basically traced by someone on a computer and then highlighted in green. And if we remove the imagery, this is top down, if we remove the imagery, we can see that the corals are all one species, multiple genotypes, so multiple genetic individuals. So we do have genetic richness in this field, but it's one species. And they're not exactly uniform, but they're relatively uniform through this thing. And so that's what we've been doing for a long, long time. Now we've graduated and kind of looked at something that looks a little more like tropical farming, where we want to have multiple species in these assemblages. We've recognized that not all corals are the same, and actually the interactions between those corals are very important. So we've added a bunch of different species to these things, but it's still in a relatively uniform distribution and not a very natural assemblage. And what we're going for now, where Mote is headed in this kind of innovative space in our ecological approach to restoration, is a more natural assemblage where we've got many species, not just a handful that are relatively easy to work with in nature, but some of those ones that are rare, some of those ones that are uncommon, some of those ones that are really common, all of those corals that we've ignored for one reason or another are important in that ecological engine that we're trying to rebuild on the reef. And we don't want them separated into little neighborhoods around the reef. We want them all intermixed. And one of the reasons that we didn't do that is because when you put corals next to one another, they do what? If they grow and they're two different species or individuals and they touch, they fight, right? That's competition. But that's not a bad thing. If we get to the point where we have competition among the different critters that we're putting on the reef, we win. So we're trying to take a bold step away from the norm and try out some of these natural assemblages on the reef. And the result is that you get this non-uniform distribution of many different species and genetic individuals within each of those species. So it gets very, very difficult to keep up with the logistics involved in this. Is you have to grow many different individuals. You have to know that they are different individuals, and then keep track of where they are in the nursery. And these guys can attest to that's not easy. And then you have to pre-plan where they're going to go, how you're going to orient them on the reef, and why. We use large area imagery, just as in the other one. And this is an early iteration of that multi-species assemblage down off of Key West. And you can see if we remove that, you see that now we have multiple different species. The different colors are different species of coral, all intermixed with one another, so that you know that you don't just have the blue guys over here and the orange guys over here and the yellow guys over here. They're all intermixed, and they're relatively they're not evenly distributed. They're definitely patchy, and it's a much more natural distribution of individuals in the nursery. And the idea is to overcome those allele effects. So if you have these natural corals on a reef, purple might be able to reproduce, right? They can't move. They're just going to spit their goodies up into the water and hope that they meet in the water column, produce a larva, and then somewhere downstream that larva recruits. But the green guy, he or she is definitely not reproducing with anyone. Neither is the red, blue, or yellow. We want to go in there and install a bunch of additional genetic individuals into this population, and we hope that and we've designed these clusters so that every one of these sets of plugs is going to fuse and become a reproductive individual. So now all of a sudden the red guy or girl or hermaphrodite will have multiple different individuals that now the probability that red species is going to fertilize larvae and reproduce goes up dramatically. So we're doing this to overcome allele effects because we do not want to put all of the coral that needs to go back out onto the reef on the reef. We want them to do it. We just want to very thoughtfully and strategically place these spawning hubs or these reproductive communities in places where they will then take over the role of restoration, and their babies will restore the rest of the reef. Part of that involves not just can we put corals onto the reef, have them survive and reproduce. Yes, we can. We've demonstrated that repeatedly for many years. We can definitely do that. How do we then restore the ecosystem, right? So I very intentionally do not, it's not on here. I am the program manager of coral reef restoration research, not coral restoration research. And that is very intentional because yes, coral are the foundation species. There's a whole community that are supported by that foundation. And one of the things that we're looking at is what I was getting at with the forest analogy, is that if you clear cut a forest and you just leave it alone, it's not a forest that comes back immediately. It goes through a series of ecological successional steps, right? The first thing that comes back are weeds and rushes and grasses and things like that. And then eventually you get shrubs and bushes. And then you start seeing trees. And then through time, you get different trees and bigger trees and so on. And eventually, long down the forest, long down the timeline, you get what's called a climax community, which is a forest, sometimes. But what we're trying to do is right now on Florida's coral reef, we've gone from something like 60% historic cover. 60% of that reef was covered in living coral tissue at one point, to less than 1 in most of the reef. So we have a dramatic shift in the ecological community. And with trees, you've got lots of seeds. They're very fecund. Those seeds are actually very effective and very, very successful at recruiting to new places. Just ask my yard. Coral propagules. One, You have to have enough of those individuals in close proximity to one another for those larvae to actually be fertilized, and then they have to survive, and they're not very good at that. And then they have to have a bottom that is appropriate for coral larvae to land and recruit, land, settle, metamorphose, and survive to recruit into that population. We don't have that right now. There's a whole bunch of those steps that are broken before we can complete that loop. So we need to address all of those things, and ecological succession is one of the ways that we're doing that. Instead of going straight from clear-cut forest to redwoods, let's start with grasses and brush and bushes and things like that and start to rebuild a lot of those ecological interactions. We need to stabilize the reef framework. We need to repair a lot of the carbon and nitrogen and detrital cycles that have been broken because of all of the biomass that we've lost. And one of the ways that we're doing that is with a whole bunch of material that we get from coastal construction projects, from the Florida Department of Transportation and a number of other groups. A lot of those groups need to replace bridges and things like that. And on those pilings and on those seawalls and things like that, there's a lot of coral. And it's all species that most people just ignore in restoration because they're doing very well, right? Like when you're restoring a forest, you're not really looking at grass as a species that you really want to focus on. Same thing has happened in coral reef restoration. We're taking a closer look at these species to repair some of the ecological interactions that facilitate higher order species. So in other words, we're going to plant grass to get redwoods. And we're working with a group from the U.S. Geological Survey, Lauren Toth, and then Jim Hench from Duke University, the same guy that I helped do some of that wave energy mitigation, because we're looking at not only the biological and ecological framework there, we're also looking at the geology of the reef. Can we stop it from eroding and keep that ecosystem service that it's providing for our insurance policies? And then also, excuse me, can we look at how that restoration effort affects the roughness of the reef and the hydrodynamic environment? Do we need to place these things in very, very strategic ways so that their propagules are going to land where we want them to? So if we want to restore this area naturally with recruits, where do we need to place our restoration efforts to let them take that effort over? And Jim and Lauren are doing a lot of that hydrodynamic modeling with us. And sorry, there's a lot of tree analogies in this one. So this is to highlight some of those multiple series of successional and facilitation cascades. So everybody's familiar with the live oak, and then that stuff hanging from it is Spanish moss. That's actually 2 levels of foundation species. So the live oak is a foundation species. It provides architectural complexity. It allows birds and lizards and all kinds of stuff to live under it that couldn't live without that tree there, right? It provides shade, it provides structure, it's got all kinds of bugs and everything. And then when you have Spanish moss come in, it increases the number of species that can use that space by holding in moisture and humidity, by creating even more and finer architectural complexity, facilitating a whole suite of other species. We're looking at coral reefs in the same way, but they are infinitely more complex. One of the other issues, we talked about the U.S. Geological Survey and erosion. This is at hens and chickens, just that way. What this is not a pillar coral. This is where the U.S. Geological Survey I don't remember what year they actually made the core, but from time to time they study the geologic history of reefs. And they do that by taking a core through the reef, because the reef grows by iterating, putting calcium carbonate on itself, and it's laying down layers on top of layers on top of layers. And you can drill down through that geologic structure, pull that core out, and read what was going on through time, almost to the day, through geologic history on these reefs. And they take these cores all over the place, and when they're done, there's this gaping hole that can be 20 meters long at times. So they fill that hole to avoid it becoming an erosion, fomite, with cement. Cement does not erode nearly as fast as calcium carbonate, the reef structure. This is one of those cores that was taken at hens and chickens 17 years before this photo was taken. And you can see how much of the coral reef has eroded in that time. We've lost, and that is, I mean, you can see here's an adult ecanthurid. That's a fish that's this big. That post is about that tall. So we've lost across the entire reef structure that much calcium carbonate just to erosion. If we lose that structure, obviously that wave energy that's coming in, that 97%, is it's going to decrease, right? More and more of that wave energy is going to get through there. And the reason that we have this is that erosion is always happening. Accretion is the other side of it, right? We can't stop erosion from happening, but we can slow it down and build up the positive, right? And coral reef growth is kind of a balance between those things, erosion and accretion. If we have enough stuff out there building more bricks onto the structure as they're being eroded away, as long as we have more stuff putting calcium on than stuff taking calcium off, the reef is going to grow. Obviously, we're not in that situation, especially here at Hens and Chickens. But can we use high-density coral outplanting to not only increase the accretion side of it, we lock up that surface that is being acted on by erosion under live coral tissue and basically rapidly adjust the situation where you have a growing reef, or at least one that isn't eroding as fast. Can we do that with outplanting at very high density? We haven't done this in the past. We've never tried this. No one's done this around the world. We've looked at placing a few corals here and there. Now we're going to look at, can we take this plot on the reef from 1 to 5% cover to 60% cover very quickly, and then what does that do to the accretion budget on the reef? And again, we're working with the same group of people. It's actually part of the same big project. And then there's another tree analogy. There's another series of really cool ecological interactions that we're looking at, because we've only been talking about the bricks that we're using to build the coral reef structure, the corals. We haven't talked about all the other critters that are facilitating things. So when you build a coral garden, or well, when you build a garden here, you deal with weeds. Does anybody have a garden here? And or Maine, yeah, for sure. Yeah, in the spring and summer, you'll have all kinds of crazy stuff show up, and then it grows even faster than it does here. Basically, this is another facilitation cascade. So this is an acacia tree that produces a resource that ants use, and the ants actually guard the acacia tree from herbivores. On the reef, we're looking at a very similar interaction between species that, we're putting out corals, and in our coral garden, there's going to be weeds. We need those weeds, right? Algae. We need algae on the reef, because it's the only way that we take solar energy and put it into things like fish and crabs and urchins and things like that. The only way that you can take solar energy and put it into animals is if it is used for photosynthesis first, becomes a sugar, and then is eaten by something else. These are the critters that we're putting out there. One, we have way too much algae. And when you do, so you need it, but you need it to be eaten by something, and then that thing to get eaten by something for that energy to travel through the system. Right now, we have way too much of it. And when you have way too much of it, they don't stop producing those sugars that I was talking about. They just aren't getting eaten, and so they start dumping that sugar into the water column. That sugar is food. It's fuel. It's fertilizer for microbial communities. And not all of those microbes are good microbes, right? And if you dump enough sugar in where you have lots and lots of algae in a system, and you keep dumping sugar into the water, eventually you're going to get something that is not, a beneficial microbe at a sufficient density that you start seeing disease show up. And then that is going to kill off more coral and make more room for algae to grow. And then you're going to have more sugar in the water, and so on. So this is a vicious cycle. And to do that, skipping slides all over the place, we're looking at two unlikely critters. One that everybody might know, the long-spined sea urchin, Diadema antillarum. It was very common here until 1983. And then there was a disease that went through the entire Caribbean region, and in 13 months killed off 99% of these, from the Carolinas all the way down to Brazil, all the way over to Africa, and all the way out, all the way through the Gulf of Mexico, Gulf waters. wiped them all out, or almost all of them out. They started to come back in the 90s, got wiped out by the same disease again. They started to come back in the early 2000s, got wiped out by the disease again. Started to come back in 2022, and that disease showed up again. And we finally identified what it was, almost 42 years after it was first observed. It was a ciliate that has now shown up in another population of these guys, a different species over in the Mediterranean, and now is in the Red Sea doing the same thing. That was one of our keystone herbivores. So everybody knows what keystone means. If you remove it, everything else collapses. Or so we thought. And keystone species are good, right? That third word in the title was resilience, right? So if you have a keystone species and a disease comes along and kills it, that's not very resilient, right? So keystone species are inherently not a resilient factor in the ecosystem. And so, while we are absolutely putting a lot of effort into restoring this critter that eats algae and in other words, weeds our coral garden, we're also looking at an unlikely character, this crab. And this is the Caribbean king crab. It's the largest crab in the Western Atlantic. They eat algae, and they eat lots of algae and algae that other stuff won't eat. But there's very, very few of them. So we at Mote are working very hard to increase their numbers. In other words, we've got plenty of colleagues working on putting more urchins out there that will eat the salad on the reef that's fighting with our coral, but then we also want to increase the stability of that system by increasing the biological and ecological diversity. So we're also looking at putting out these crabs. This is a project that we did where we plugged a whole bunch of these limestone tiles, actually like right out, probably right out here, Pickles. I don't know where I'm at. Pickles Reef. Yeah, there we go. Thank you. At Pickles Reef for quite a while, about 18 months. And they grew this very, very natural kind of assemblage of algae that you find out on the reef. And then we put in these teeny tiny, so these are not, well, that's a picture of the crab that was on there, but this one is not. You can see the little crab is right here. The little urchin was, teeny tiny, these cultured critters for 2 weeks. And you can see that the difference between the control and the ones with the two critters is dramatic. And basically this highlighted that not only, and I'm missing two treatment groups here, where there was no herbivore, urchin, crab, urchin and crab, urchin, crab. The one that did the single best was the urchin and crab. So you again have diverse assemblage of these critters, becomes a more stable system and actually transmits that energy and the algae up through the system much more effectively. We've got plenty of colleagues that are doing this one. We are working at Mote on the Caribbean king crab, and we're doing it in bulk. This is some of the team up in Sarasota at our very, very large Mote's Florida Coral Reef Restoration Crab Hatchery Research Center, which is dedicated to mass producing this species to facilitate our restoration efforts. Here's a picture inside of that facility. These are 25 feet by 25 feet by 5 foot deep tanks. And they've got, right now, there's 3 of them. 2 of them have crabs in them, and then there's a whole series of systems out here. Right now, there's about 800 crabs in that system, producing somewhere between 10 and 15,000 or very early stage juvenile crabs almost every month. We're still working on how to produce the algae that they eat, which actually seems to be more difficult than producing the crabs that we're trying to. But one of the bottlenecks to using this, and the idea is to produce these crabs, take the little ones out, put them out alongside our corals. They'll eat the algae so that the corals survive longer, grow faster, and reproduce sooner. One of the bottlenecks to this is a regulatory bottleneck. Same thing with diadema, same thing with the urchins, is we have to have a veterinarian certify that these crabs are healthy before we can put them into the reef ecosystem. It was relatively difficult to find a crab vet, but we did. And so one of the things that we've done is go through and sample a lot of the live wild crabs in each of the different ecotones or each of the different geologic regions throughout the Keys and up the east coast of Florida. And we wanted to look at, are there any things inside of these crabs in the natural population that aren't in the cultured crabs that we're producing, or things that are in the cultured crabs that we're producing that aren't already in the natural population, which would signal something that we really need to pay attention to before we start putting them out onto the reef. We don't want to introduce things just like with the coral. Don't want to introduce diseases, don't want to introduce parasites, don't want to overdo any of those kinds of interventions. And all of this, the coral, the ecological succession, the erosion, all of these kinds of things, along with the crabs and a number of other ecological interventions that we're doing, are all going into a very large, landscape level experiment where we are working at 10 different reefs up and down the Florida Keys. Each of the ones that are labeled in white here are the 7 Mission Iconic Reefs locations, and then there's 3 blue ones, American Shoal, Coffin's Patch, and then Ham Reef off of Isla Morada over here, that are non-zoned, right? So each of the white ones you can't fish at. Each of the green ones you can. So we want to look at how do these interventions affect, reefs where you do have a lot of legislative protection, and then how does this work on reefs where people can do the things that people like to do on reefs. And in each of those locations, we've set up a series of pins, so we have fixed plots. So they are 10 by 25 meters in size. And then on those, got a series of plots here. We're going to not just, outplant multiple species and multiple individuals within each of those species on these plots, like I was saying, and then never come back. We're going to iteratively, annually come back and install additional corals in those species and additional species through time. So this is yet another departure from the norm where we go out and restore an area and then we walk away. That's not actual restoration, that's outplanting. And we want to restore the ecological complexity. So we're not just visiting a place and then never coming back. Now we're iteratively outplanting and increasing the complexity of those interactions through time. And then on top of that, we're going to install a bunch of crabs to weed that coral garden. And we're going to monitor those, their effect on the fish community, their effect on the algal and sponge communities, and how all of these things interact to drive patterns outside of the plot as well. So we want to see, by doing this over the course of several years, do we start to see corals recruiting outside of these plots? So is the plot the bounds of what this intervention is, or is there a shadow that's well outside of that? So in other words, do we need to put corals in a 10 by 25 area to restore a 50 by 50 or 100 by 100, or do we just need to keep doing that down the line? A lot of this involves an enormous amount of logistics and planning, and part of that is using things like satellite imagery to start doing some pre-planning of where we're going to site these plots and where we're not going to site these plots, using a lot of eyes on the reef to ground truth yes or no. So that's what these green and red ones were. The red ones were not where we wanted to work, green ones were good. And then go out and use large area imagery, and you can see there are some corals on this plot and these little tiles that tell us what's going on in these photos. And from that, we're building these digital tools where if you put all the corals on this plot and then you leave and then you come back and you put more on there, which ones did you put in year 1 or 2 or 3 or 4? So you have to build a digital map of that plot that the people in the water can actually follow, so you can track individual critters through time. And all of that relies on very effective and very efficient digital tools. And part of one of the other kind of innovative avenues that we're going down is building some of these digital tools that make doing all of these things a little less complicated, or at least much more precise. So this is a workshop that we just hosted at Reef House Resort and Marina with members of all of the regulatory agencies in the area and all of the practitioners, where we came together trying to build out a series of tools that allow us to talk to one another in the same data language and do it seamlessly and do it more efficiently, so that a task that used to take weeks in terms of picking corals and sending them from one place to another now takes minutes. And part of that is also innovating in Mote's nurseries with our partners at Sea Foundry, which is a tech startup from California. There was a bunch of guys from Google and Tesla and a couple of other places that got together and decided to use their black magic for science and for conservation. And this is one of our staff members at the Key Largo nursery standing in front showing two of our partners from TNC and the University of Miami the robot. And you can't really see it. There's a series of metal apparatus over this thing that is a robot that takes an end effector with a camera on it and can go through that raceway and take a photo of every single coral multiple times a day or a week or a month. And then, more importantly, on her phone, she's showing them the app that our colleagues have made that makes something as benign as inventory management. Where is that plug of coral? Who is it? What species is it? What genotype is it? How long has it been here? What happens if it dies? What happens if I fragment it? All of that information used to be pen and paper, but when you're talking about 150,000 fragments of coral across 5,000 or 8,000 different genetic individuals in 3 or 7 different locations, and that number and those stocks are changing every single day, sometimes multiple times a day, and those corals are going out into the field or between sites, all that becomes infinitely complicated without a digital tool. And now we've got some things that were relatively simple when you think about it that are transformative in terms of efficiency and making that money go even further with no real change in the effort. We're then taking all of that and we're going outside of Florida. So one of the really cool projects that we're working on right now is doing almost everything that we just said, but in Puerto Rico. And that may not sound really exciting, but I'm super excited about it. I like going to visit that program, and there's a whole different regulatory environment. The ecological environment is incredibly different, and all of a sudden you don't have a continental reef where, obviously here on Florida, if you want to get into deep water, you have to go several miles out. They have to go several hundred yards from shore to get into the same kind of deep water, and then they get really deep. So it's a very, very different ecological environment. And the importance of this is not that we're working in Puerto Rico and that's really cool. It's that you expand what's called the inference space. So if we do this work here in Florida, what we can say is this works in Florida. If we do this in Florida and Puerto Rico, we can say that this works on continental reefs and on insular reefs and across most of the Caribbean Sea. So this is a very interesting, very, very dramatic kind of increase in the power of the science that we're doing. And we're also replicating these experiments in a number of other places that we're not just there yet. But beyond having these professional partners, restoration is obviously a team sport. We have lots and lots and lots of scientific and professional partners, but we also need people like you to get involved in these things. So how do recreational divers or interested people get involved with the science and restoration that we're doing? You can visit things like Scuba Pro, which is a vendor or a brand that we have right here in Divers Direct. They're a partner of ours, and we offer a number of different opportunities to get out and work with the team in Key Largo or down in Summerlin Key to produce a bunch of coral, learn that process on land, but then also participate in the restoration and propagation and so on processes in the water. We've also got our colleagues at iCare in Islamorada, where we have a partnership with them where you can absolutely get out and experience the role that restoration practitioners play in putting corals back onto the reef. We have a very wonderful volunteer program that I think that you guys could talk to Aaron and Katie about, and we have plenty of opportunities for people to volunteer their time and effort and get involved with the science that we're doing and in the actual interventions. And then you can visit all of these facilities. So the main facility at Mote's Elizabeth Moore International Center for Coral Reef Research and Restoration, or IC2R3 down on Summerlin Key. Mote likes to put really long names in front of every facility that we have. You can absolutely visit that campus. You can visit our Isla Mirada campus and our Key Largo campus if you'd like to tour and see some of these corals in action. The robot is still there at Key Largo, and I'll bet if you ask nicely, Preston will make it do stuff. You know, it doesn't make R2D2 sounds yet, but they swore that they're going to make that happen. You can also get some hands-on experiences even if you're not a diver. So Slice of Paradise is a program where you can actually participate in producing some of the microfragments that we turn into, larger colonies on the reef. And those corals are absolutely used in our restoration efforts. And if you would like to do that, you can absolutely do it at Mote's Key Largo Nursery, the Isla Mirada Nursery, or now I think down at Summerlin Key as well. And then one of the ones that I love doing is the Protect Our Reefs license plate. You can get this specialty license plate, and $25 of your fee goes to Mote Marine Laboratory. And most of that money goes directly into supporting Mote's coral reef restoration research and outreach and education programs. And not only does this support Mote's science, but we hold a competitive grant program where groups from all over the world, I think you have to be a state, or at least partnered with a state institution to get one of the grants. But we have a competitive request for proposals where people propose their project to us and we award them money. And we've awarded millions of dollars over the last few years and supported some really, really amazing science to various scientists throughout the United States. So getting yourself a Protect Our Reefs license plate directly supports our efforts to restore Florida's coral reef. And then you can obviously donate if you wish. So restoring coral reefs in our lifetime is not science fiction, and it really isn't. And we've seen the evidence of we don't need hope. That's nice and fun and everything, but we don't need that. We've got science, and our data are very clear. We've had quite a few failures, but that's science. However, the successes are glaring and they're amazing, and we're building on them every single day. And one of the things that makes that go is financial donations and funding. So among all that, I have to thank all of the amazing people that work on this work and with me over there at Mote. So Lower Keys, there's a group over there. The Upper Keys restoration team is here. Our grazers research and production teams in Sarasota and in the Florida Keys, and then our amazing community engagement, education, and volunteer teams at both in the Upper and Lower Keys are all fundamental to making this work happen. And with that, I think I'm way over time, but thank you. And does anybody have any questions? Yeah.
Yeah, is there an entity that oversees and coordinates the work of Mote and Coral Restoration Foundation and Reef Renewal and iCare?
Well,
Or do you guys
yes and
so you don't
no.
step on each other's toes?
Yes and no. So we're all participants in Mission Iconic Reefs, which is a NOAA-led initiative that works in the Florida Keys National Marine Sanctuary at those 10 reef sites that I was pointing out. And we're all three working in the same place, all 5, 6 working in the same place at the same time. And they try to do a little bit of coordination there. But a lot of that coordination, a lot of those logistical conversations and deconfliction, conversations on all those kinds of things are conversations among those partners. Restoration really is a team sport. We all work together very closely. And I speak with Fanor at CRF and Ken at Reef Renewal and Kylie at iCare and all of those different, I'm married to the one at the Boy Scouts. So we work with those groups very, very closely, and we're in relatively close communication all the time. So there's quite a bit of coordination among us. There's also quite a bit of competition.
Sure.
Yeah, and we're better, but and I tell Fenor that all the time. But a lot of that coordination is done on the ground. Like we are actually coordinating a lot of that among ourselves. However, there is NOAA, out of that, at least from kind of a 30,000 foot view in some of those locations. And then FWC does a lot of that coordination through our permits. So all of us have a series of state, federal, and local, and regional, and other federal permits. There's like 6 or 7 different agencies that manage this area from a regulatory perspective, and we have coordination through all of them all at the same time. So there's coordination all over the place, but the vast majority of what you're asking is done just between the groups themselves. So it might look really, really competitive, and it is, but it's also very collaborative. So it's a great, very supportive community to be part of.
So Jim Britton found that the urchins that they raised in the lab
Yep.
didn't survive very well in the reef because they weren't just having to stick onto things.
Oh yeah.
So they have those reef
I think
balls.
that's much less.
Like other crabs, do they have problems going from lab to reef as
We
well?
don't know yet.
Okay.
We don't know yet. We don't think so. But that's a problem with any stock enhancement program where, even fishes. When you aquaculture something in this like coddled environment with no predators and everything, they're real stupid when you put them on the reef. And those urchins, it's not so much that they're not used to sticking to things, is that they're real dumb. Yeah, they're not worried about predators when they really should be. So one of the things that we've worked with the crabs, or at least that we're playing around with, is behavioral modification. So if you scare them, a bunch of times, then they'll be scared of stuff. And when you put them out on the reef, they're not going to like reach up for food and get eaten. So it was a really cool project, or it is a really cool project, where we worked with one of the local, was it Ocean Studies School, with these elementary school kids, and they made these puppets. And it's like a dish glove that they put googly eyes on and doily, all this kind of stuff. And it was a shark and a turtle, and the typical predators of crabs. And the idea is that you present a visual stimulus, and then you mess around with the crabs, and they associate a visual cue with a physical cue. And so when you release them, if they see something, they run away. Right, yeah, the idea is pretty cool. We don't think that their eyes are actually, accurate enough they can't tell the difference between a glove and a brick and a scuba tank, but it's a really cool way of integrating youth into the science and actually thinking through kind of behavioral ecology and things like that. So that is something that we're very concerned about. It is something that has plagued our colleagues with diadema, those urchins, for many, many years. And that's not limited to the cultured urchins either. If you take urchins from this reef and move them over here, they all get eaten. That is a species that has been a frustration for so long, and we feel for them, but we are starting to crack the code. The community is starting to crack the code on those critters, and it's one of our very close colleagues, Jim Britson's major advisor now, Josh Patterson, and Jim are both working with us on a series of projects where we're looking at crabs and urchins together as complementary species, and they're starting to make some really good progress.
So going back to that coral colony that we outplanted like 10 years ago, that's about to turn 10.
Uh-huh.
So we obviously know that fragging increases the rate, I don't know, like tenfold, whatever.
Yes.
Do we know, or do we have any data of how it's growing in the water? Like, is it still an increased rate compared to
Yes.
natural?
Yes, we haven't actually measured those colonies. I mean, they're like this big. I was out next to them like 2 months ago. And it's not just one. This wasn't just one colony. This was a series of about 17 clusters that we put out in 2015 that fused, and most of them spawned in 2020. Now, next week is the spawning window, which would be the 10-year anniversary. I'm hoping that they go in August because I'm not going to be here. But those colonies, obviously, when you make those microfragments, when you cut the coral into very small pieces, that accelerates the growth. Just like when you cut your skin, it goes into that healing mode and grows much faster than it does right now. The same thing happens when you microfragment corals. Their growth goes up by 40 to 50 times, and it maintains that rate until they are healed. So if you can keep that laminar growth going, they'll actually grow very fast for a long period of time. Once they fuse, they kind of slow down to the normal rate of progression and start that kind of three-dimensional growth as well. It's still faster than if you started with one, but no, those colonies aren't still growing 50 times faster than they would, But it doesn't matter. They're now reproductive. They're putting millions of gametes into the water column every year, hopefully. Go ahead.
So you said you went from planting the same species to more of a biodiversity.
Yes.
Are you noticing that because you're doing the biodiversity, your success rate or your rate of growth is lower on those coral reefs?
It's definitely not slower. Like we've done each of those species in isolation, and even kind of some two species kind of clusters and everything. We haven't gotten enough time since we started the multi-species assemblages to say one way or the other. However, on one of those sites that I actually had the image up here on, it was a nearshore site off of Key West, just off of Higgs Beach, about a half mile offshore. Those corals were outplanted right as the 2023 heat wave hit. And in fact, the next day, some of those corals started bleaching. But even through that devastating event and through the second one, we had something like 70% survival of those corals. So that was dramatically higher than we were expecting. That was dramatically higher than a lot of the other species and assemblages throughout the reef tract. So we've definitely seen some evidence that this more rich and new mode of outplanting using kind of ecology is absolutely potentially having some facilitative effects. And then there's numerous examples in the literature where multiple species facilitate one another through time. But we should start getting a lot of those data that'll actually give us kind of definitive answers to that question here in about 3 or 4 months. We'll start getting those data in.
So you talked about a heat wave that came through. Are
Yeah.
you seeing a gradual increase in temperature? I'm thinking.
I wish it was gradual.
Like
Yeah.
in the Gulf of Maine, where it's one of the most rapid
Yep.
heating
Yep.
bodies of the ocean right now. We're seeing species come in that just do not
Probably belong here.
Oh,
Yeah.
Well, yeah. I mean, at the very least, Long Island.
Yep.
They're
Yep.
showing up.
Absolutely.
So what's the predictive modeling say for what's going to happen here relative to then what species may or may not move in and impact it? I just don't know the water.
So it's like two questions there. Like, so one is like the latitudinal range expansion, right? So the equator is kind of typically the tropics, and the tropics are based on all of basically the incidence angle of the sun. And normally that defines kind of thermal limits of things. Like tropical species they get into subtropics a little bit, like here, but beyond here, you're not really supposed to get much. Fort Pierce is where coral reefs can't really go beyond, or so latitudinally. As that sea surface temperature continues to increase through time, that latitudinal limit should start to expand. And you should start seeing other things. And you can already see it if you go to St. Augustine and you look at the salt marsh. So up where you guys are, you have those salt marsh with the grasses and the little snails and all that kind of stuff. You get that all the way down to about St. Augustine. And then from there down, you start seeing mangroves. That replaces that kind of coastal wetland area. In St. Augustine, you can see that ecotone, where it is starting to expand north, where you start seeing that spartina, the grass from the wetland, and black mangroves, and eastern oysters, and tropical sponges, all in the same spot. And every year that moves just a little further north and a little further north, you'll start seeing the same kinds of things in obviously subtropical areas, and then you'll start seeing species not being able to handle the conditions, without adaptation and everything. So you should start seeing some rapid evolution, and we've seen a lot of that in corals, where you have these thermally tolerant corals that we actually really focus on in terms of putting out the most resistant and resilient individuals. But you also start seeing other things get facilitated, like increased disease incidence and the emergence of new diseases and things like that. In those more temperate locations, it's a little more insidious, where obviously you'll start seeing large conspicuous species show up that wouldn't be there normally, and large conspicuous species that are supposed to be there not there anymore as they move out into more favorable conditions. But you also start seeing primary productivity. So, you've green water up there from time to time through the year, seasonally. Those seasons are going to start getting longer, and that means that you're going to start seeing things like dead zones and things like that as those algal communities, those little tiny microalgae boom and bust and use up all the oxygen. And so you'll start seeing all kinds of like weird ecological effects in those areas if that condition persists through time. And it just becomes much more fragile. So you're starting to change the definition of what's adaptive in those locations, and you'll start seeing all kinds of unintended consequences.
Crazy question. So when you fragment coral, they grow a lot quicker, right? 'Cause
Yep.
you're agitating the outside of them. Could you not go to those colonies that you outlined and just trim the edges and let it kind of, again, reboot that 50 times growth rate?
It's certainly possible, but you'd also be opening up a wound in an area where there's a lot of microbial activity going on in a place where you can't manage those interactions. So it's entirely possible. And there is a series of methods, I call them chop and drop, where you go and outplant particularly the branching species and you let them grow up into a pretty good sized colony. And then instead of pulling some out of the nursery, you just go to that one and chop a bunch of pieces and plant them next to it. And then they grow, and then you chop a bunch of pieces and grow next to it to build a thicket. And that works beautifully. It absolutely works, and it does exactly what you're saying, is you damage not only the parent colony, it starts growing faster, but you make a nubbin and it's growing faster, and you plant it next to it and vegetatively propagate it. With the massive species, it's a little more risk of disease and predation and things like that. But it's entirely possible. I've definitely seen people microfragment corals underwater on the reef and even damage the edges of living colonies to try and get them to grow faster over a bioeroder or things like that. And it works 30% of the time, it works every time. Yeah.
I have 2 questions.
Oh, all right.
The first one is, so because we live down here, we do see, I do see how some of our corals in the park and out of the park is damaged. We have a lot of tourists who don't know that much about it, and they do stand on them. They do all these. So I do know
jerks.
that we do have some. I do know that we have some coral damage and coral loss. So how? And I know that you're being successful restoring the coral that you're growing, but how is that in rate of how much we're losing? Are you still successful? So that was question number 1.
So question number 1 is,
Your rate to restore
is our rate of restoration keeping up with the rate of degradation? Absolutely not. That's that whole, we need to expand what we're doing at this scale to this scale. And that is a major research objective for Mote in particular, is how do we take what we're doing at this fine scale and bring it to a scale that's ecologically meaningful for a reef that is 360 miles long? We don't have the answer to that yet. A lot of it right now is 29-hour work days for these guys and like no breaks, and you guys need to work faster, you need to be using 2 saws at the same time and just like go. No, and those are the questions that we continually ask ourselves, and we have to think outside of the box. It is not pushing these guys to their limit and running 4 saws at the same time, and no breaks, and that's not the answer. It is how do we innovate in the materials that we're using, the not just the physical materials that we're using, but also the biological materials that we're using. We need to be using corals that survive longer, grow faster, resist disease, resist thermal stress, all of those kinds of things, but then thoughtfully placing them in places where they are going to take over that restoration action and amplify it by downstream recruiting new corals that are then eventually going to grow into new colonies that are going to produce babies and restore other areas. So this is not a short term, this isn't a thing that we're going to see in a very short brief period of time. This is a long game. And it's a very high activation energy where there's going to be a lot of slow and then it'll start to grow, and then it'll start to be an exponential increase if that rate of stress doesn't continue going up at the same rate. So it's a balancing act, and it's something that it's a question that the answer is always changing.
And I feel like as Florida residents, every Florida resident should be also more educated on the topic. I don't think there's
I agree.
enough education out there about it. Which comes to question number 2. So I am a teacher at Coral Shores High School, and we do have a group of students who do work and volunteer with the Key Largo Foundation. But those are our marine science students, which is a very small group out of all of our students. I'm also in contact with Marathon High School and Key West High School. And every one of them wants to always know how can, we get more of you guys to come and do presentations to not just our marine science students, but for example, put you in our library, put you in our auditorium, so we can have a larger scale presentation.
So how do we get the word out?
Correct, information.
Bigger,
Exactly what you
faster,
said,
longer,
yes,
yeah.
volunteers, the
I think you
time.
talk to them. But yes, we do presentations like this all over the place. We're happy to do those kinds of things. Preferably not on holidays.
We are not open on holidays.
You're my favorite one. Yeah, like, yes, yeah. Although we are absolutely willing, and we can cater those kinds of things to younger audiences and do things like puppets and talk about how
A
that.
lot of times, younger audiences also teach their parents, so it does help.
Oh, absolutely. Yeah. And
In the upper end of the Keys, we have full-time educators on
Yes.
staff with Mote, who can lead programs themselves or facilitate, our scientists coming in and interacting with our students.
and they're way better at it than me. Yeah. Yeah.
I know at the end of the month, we're doing talking about the range we can provide. We're doing a program at St. James Children's Center with our high schoolers. So we really, truly start all over with our littlest ones here after our high school. Thank you.
Do a couple questions for the giveaways.
Oh, okay. Yeah. So they've got a couple of giveaways. And
Oh, is that cooler one to do?
we just start with any one of them. Just start with any one. Does anybody remember? Yeah. How about like closest to the answer gets this one? Like, how many corals has Mote outplanted on Florida's coral reef since 2008?
258,000.
Anyone? No, I think that was it. Yeah. That was almost exactly it. Good job.
I hope they work for you.
I know. I'm like, all right. No more of you guys. Florida's coral reef extends from Fort P. Oh, no, that's not. That's a silly one. No. That'd be beyond that. Yeah. 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 How can you get involved?
And volunteer. Volunteer, I plant. And
What's that?
I go with eye care and I do planting and maintenance.
Wonderful.
So getting involved.
Yep. There you go. Thank you. And then the final one.
I shouldn't be getting this.
Yeah. We talked about a crab. Yeah. Yeah. What's special about its size?
It's the largest
Where? In the Caribbean? Ah, yeah. Who had it first? Oh man.
I think Daryl.
Yeah, Daryl got it. Yeah, there you go, Daryl. All right. Well, thank you guys. And now the cool. I'm kidding. Yeah.
Let's speak of showing up in lobster traps in Maine.
Yeah. Well, thank you all so much for coming. And if you have any questions, please don't hesitate to reach out to me or to Aaron or to Katie.
See,
Thank you.
folks in the business school.